Hi all, I'm Malavika, and in this video I'll be explaining the production of monoclonal antibodies using the Hybridoma technique. So what are monoclonal antibodies? They are antibodies which are derived from a single clone of plasma cells and they act against a single epitope of an antigen. The Hybridoma technique of production of monoclonal antibodies was introduced by Kohler and Milstein in 1975. The principle behind this technique is that B cells which are stimulated against a single epitope of an antigen will be fused with immortal cells to produce hybridoma cells. Hybridoma cells will produce monoclonal antibodies of the same antigen specificity. Coming to the steps of hybridoma technique, firstly, a mouse will be injected with an antigen which bears the desired epitope. After some time, the B cells from the spleen of the mouse will be collected. Next, we need immortal cells. An example of such a cell is myeloma cell. Myeloma cells are cancerous plasma cells that closely resemble the B cells of mouse. Now, myeloma cells are found to have the ability to produce their own antibodies, and this property should be eliminated. So, myeloma cells are genetically modified with two mutations to obtain double mutated myeloma cells. So they lose the property to produce their own antibodies, but they retain their immortal property. The next step is fusion of the splenic B cells and mutated myeloma cells. This fusion takes place in polyethylene glycol broth. After this, three types of cells will be produced: unfused myeloma cells, unfused splenic B cells, and fused hybridoma cells. In order to obtain the fused hybridoma cells alone, purification should be done by subculturing on HAT media. HAT stands for hypoxanthin, aminopterin, thymidine. Now, why are we using this medium? Aminopterin blocks de novo synthesis of purines in mammalian cells. So, the cells will have to perform the salvage pathway of purine synthesis. Now, the salvage pathway requires two important enzymes, namely HGPRTase and thymidine kinase. HGPRTase is hypoxanthin guanine phosphoribosyl transferase. Myeloma cells lack this enzyme HGPRTase, so they cannot grow on HAT medium. Now let me explain the fate of the three types of cells which we had obtained in the previous step. The unfused splenic B cells can grow, but they cannot survive for a long time because they are not immortal cells, unlike myeloma cells. Unfused myeloma cells cannot grow because, as I've already said, they lack HGPRTase enzyme, so purine synthesis cannot happen. Then we have fused hybridoma cells which can grow and survive. These hybridoma cells thus obtained can multiply indefinitely, producing clones of identical cells, and they can be used for the production of monoclonal antibodies. Just a note on the applications of monoclonal antibodies. They are used in the diagnosis of certain diseases by antigen detection. For example, they are used to detect HbA-Ag, which is seen in hepatitis B. Monoclonal antibodies can also be used in the treatment of certain diseases. For example, infliximab is a monoclonal antibody which acts against TNF-alpha in rheumatoid arthritis. They are used to provide passive immunity, for example, seen in hepatitis B. Then they are also used as immunotoxins that kill target cells, for example, cancer cells, when conjugated with bacterial or chemical toxins. So that's all about monoclonal antibodies and their production through hybridoma technique. Thank you for watching.